going to introduce our next speaker. James Williams is actually one of my PhD students with Fiona Watt. He's at King's College London. Many of you will have contributed over the years in the clinics to another study, which was a skin biopsy, and that was essentially to create a biobank of skin cells, which were then essentially turned into stem cells. And those stem cells are they're freely accessible really to anyone researching on the planet who needs to research into what are called iPSCs or stem cells, induced pluripotent stem cells. And they can be turned into all sorts of different organs in a dish, if you like. And so James has been working on some of those samples that, they're all anonymized, of course, that you have contributed, and he's going to tell us some of the work that he's been doing on kidneys. So James, without any further ado, I'm handing over to you. This is working. Yeah.
Can everyone hear me? Or can you hear me at the back? No.
Do you want a handheld or?
Handheld would work.
Yeah, handheld is coming. It's obviously forward, backwards.
Yes.
Yeah.
Thank you.
All right, take it away.
Hello? Hello? Is this working?
I think he has to turn it on the other end.
And now? Yes. There we go, finally. Anyway, thank you for inviting me to come and talk and making me feel so welcome this morning. As Phil said, my project involves using a special type of stem cell derived from skin samples, which lots of you people here would have donated, to investigate kidney disease in patients with Bardet-Biedl syndrome. So just to go over initially to make sure we're all on the same page of what stem cells are. Our body is made up of billions, trillions of specialized cells, which each do a specific job depending on where they are in the body. And stem cells are just the term we give to cells which aren't yet specialized. So they can become any type of cell you need in the body. We sort of think of it in terms of they can become anything. So if you imagine a ball sitting on top of a hill, a stem cell can roll down the hill in any direction and become any type of cell you need it to, as and when you need it. The problem is that in the adult body, once cells are specialized, they can't become stem cells again, so you have a very limited supply of them. So scientists like to use stem cells to figure out how certain cells or certain organs in the body develop, because they can become any cell or any tissue, and they can look at how the differences emerge between different cell types to see how the eventual tissues are formed. But again, they're quite hard to get hold of. But then 10, 15 years ago, some labs in Japan found a way of making stem cells from skin samples. So you can take a skin sample from a donor, create stem cells, then you have a bank of cells that you can use and make them into different tissues as and when you want. And the great thing about this is that when you take cells from a donor, the stem cells you have will have the same DNA and the same genes as the donor which gave you those cells initially. So if you have a donor with BBS, you can then get stem cells with BBS. And then from these stem cells, you can turn them into any type of cell in a dish. And you can look at how BBS genes affect the way you can form different tissues. So for example, with us, we can take skin cells from patients who don't have kidney disease and patients which do have kidney disease, turn them into stem cells, and as they turn into kidney cells in a dish, we can then start to compare the differences between patients with BBS and patients without BBS, and sort of get a bigger understanding of where the differences emerge and where it's causing changes, which could then hopefully give us a better platform to develop treatments and things going forward. So yeah. And then if we get the platform working well enough and we can get kidney cells in a dish that are behaving just like we'd expect BBS kidney cells to behave, then we can potentially use it as a model. As Phil was mentioning earlier with mice, we could have human cells growing in a dish where we could test potential therapies and treatments. So this is where my project comes in, and I'm sure some of you may have heard of it, and I know some of you will have contributed to it. There's a project called the HIPSI project, which involves making a huge bank of these induced stem cell lines from patients with all sorts of different diseases which affect lots of different organs. And we have 50 donors from BBS, which is amazing, which gives us the ability to test the different genes and see how they can affect the formation of different cell types. So the group in Australia developed a method to turn these stem cells into kidney cells in a dish. So on the screen, we just have some pictures of cells growing in a dish, and they basically feed them with chemicals and factors to signal to them if you imagine the stem cell rolling down the hill, they nudge it in a direction to form kidney cells. They form it into a little ball, and if you look at it under a microscope after 20 to 30 days, you start to see little tubes forming within this cell. And if you test for genes which you'd expect to be present in kidney, you see them being expressed. And you also see little tubes forming, which show the markers you'd expect to be expressed in the tubes, in the nephron, in the kidney, where you find epithelia. So you have a really good model for making kidney cells in a dish, and we have lots of stem cells from patients with BBS. So this is where my project begins. And we decided to focus on BBS10 specifically to start off with, because work that Phil and Elizabeth have done in the past has shown that BBS10 is much more likely to result in a severe kidney phenotype. So the patients with BBS10 mutations are more likely to have kidney disease. So if our model works and we're able to see differences, we're most likely to see it in BBS10. So this is just where we decided to start. And we have 3 cell lines from donors who have changes in BBS10. One we've called LIG, one we've called ZIRI, one we've called NOLS. Those are just anonymized names to help us to keep track of which one is which. And we managed to get it to work in the lab. So 2 of the cell lines, LIG and ZIRI, if you grow them for 20 days, they're able to form the different structures you'd expect to see in the kidney. And then one, NOLS, which has a much more severe change in the BBS10 gene, we actually found wasn't able to form the kidney tissues in the dish at all. So showing that BBS10 is having a significant difference and a significant effect on the ability to form these different structures within the kidney. And then if you grow them for slightly longer, then you start to see changes in the other 2 lines as well. So we just have some images on the screen showing that a healthy ball of kidney cells we grow in the lab expresses lots of markers for the tubules within the kidney, and the other 2 lines, which were previously able to show those markers, after 7 extra days of growing, they become less and less able to do so, and they look more and more unhealthy. But because we have this system, and we know that gene therapies help with this type of thing, we're able to sort of do a crude experiment where we take a healthy copy of the BBS10 gene, make it into a circular piece of DNA, and then add that into the cell, so that the cell now has a healthy copy of the BBS10 gene. So you now have these cells from donors that have BBS10, but they've got this circle of DNA which contains a copy of a healthy gene, so they now have access to a healthy copy of BBS10. And what we see is that where in the past the NOLS line, which would have had the most severe mutation, wasn't actually able to form the kidney structure properly, just by the addition of this healthy BBS10 gene, is able to look more like the healthy line again. And when you look for the markers of those tubules within the kidney, you start to see expression of those markers again, showing that just by adding a copy of BBS10 via this sort of crude gene therapy treatment, we're able to restore kidney tubules within the organoid to these BBS10 lines. We tried to publish this, but they told us they didn't believe that it was BBS10 that was causing it for some reason. So they asked us to go back and basically damage the BBS10 gene in some healthy cell lines. And then what happened was that you lose the ability to form these kidney structures again, so confirming that it is BBS10 that's causing it. And interestingly, it's still able to express some kidney genes, but it's specifically the epithelia in this model. So it's specifically the tubes within the kidney, which is where the problem arises, which is what you might expect, because BBS has been shown to affect the cilia, and the tubes within the kidney is where the cilia are most relevant. So this is the summary of that part of the book, just to show that we have this model where we can take cells from human donors, grow them in a dish, and by adding a healthy copy of BBS10 to BBS10 patients, we can rescue it, and they have tubes again in the kidney. And by taking a healthy donor cells, which normally have tubes, and damaging BBS10, we have a model where they aren't able to form the tubes in the kidney. So we have a really good model from here to look in a human background how BBS10 changes the way the kidney cell develops. And we can basically look as cells grow in a dish and start to compare the differences. So this is sort of, oh no, okay, we're fine. This is where we can sort of start to pull out differences between BBS10 and healthy lines. So this is the most important thing, I think, from the talk, is we now have a model in human cells where we can start to compare these things, and we can make human kidney cells in a dish where we can start to compare them. And just quickly, to go through some of the work that I've been doing beyond this, which might be a bit hardcore science, but we'll see, is I went through and started to actually look at what on a cellular level is changing in BBS10. So we have this structure within the cell. It's just like a long filament. It's a long fiber. It basically is called actin, and it acts as the skeleton of the cell. So it gives the cell its shape. It allows it to move around if it needs to, but it's really important for the cell shape. And we see, when we compare the BBS10 and the healthy kidney cells, we see big changes in this. And so to look at this, because it's in really small, it's within the cell, we have to grow them in 2D and look at it under really powerful microscopes. And what we see is, I'm just showing an image on the screen of some microscope images. And in healthy cell lines, we get these nice compact cells where they sort of act like tiles. They're really nice and tightly packed together. And you can see little dots forming in the center of the cell where cilia are there. And in the BBS10 lines, you don't get any cilia. And they're not nice and compact and tiled. They sort of stretch out because the skeleton has been changed. And when we look specifically at the skeleton, we can see these long fibers forming. So instead of being nice and compact, they become really long and stretchy. And this is what we sort of call, rather than being epithelium, which is what we call the tiled things, the tiled-like cells, which are lining tubules, we call it more mesenchyme, which is just another word for stretchy and not able to form like the tube around the kidney. And if we treat healthy cells with a drug, it turns it into mesenchyme, which here on the screen is going to be the cells in green. And we see it looks very similar to the way we expect mesenchyme to look. And then when we compare those cells which have mesenchyme to the regular BBS10 cells, we see that they look very similar. So we can kind of, from this model, pull out that we're expecting, okay, the BBS10 cells might actually be less epithelia, and they might be going to this fate instead. So now that we understand they're going to this fate rather than epithelia, we can maybe try treatments to reverse that and go in the other direction. And this type of change is associated with injury in the kidney tubules, so it's not something that's unprecedented. So yeah, looking at the treatments, if we try our circle of DNA with a healthy copy of BBS10 again, rather than what we see in this image of long stretchy cells without cilia, just by adding a healthy copy of BBS10, you end up restoring some cilia. You get some small dots appearing which show the cilia, and it's nice and compact and they stop stretching out. And then mimicking some work that Phil had done in the past, if you treat the cells with a really, really powerful drug which changes the cell skeleton, you can actually restore the cilia there as well and get this nice compact phenotype as well. So this is still really early stages. We haven't published this, so it needs to be investigated further. But I think it's a good example of how just having this model of human cells where we can turn them into kidney cells in a dish, we can start to look at different treatments, and we can start to understand what's going on within the kidney. So the conclusions to take away is just that we're able to use samples which have been kindly donated by people here and use them to grow kidney cells in the dish, find the differences between healthy and BBS10 patients, and then using that understanding, hopefully in the future, develop better treatments which will aid treatment in the future. And we can possibly use these as a model to test future treatments as well. So that's everything from me. Thank you. Slides which I added on the end, because you can just skip through.
I'll go on then. Yep.
I mean, should I talk about this as well, or no?
Yeah, very quickly.
So we also found changes in the way the cell divides as well, which is very relevant in the kidney. I added this on as a panic slide in case I had like 10 minutes left. So I'm going to rush through this, but we found changes in the way the cell divides, which is very important in the kidney specifically for making cilia. When you divide cells, you can reuse some of the components to basically shortcut making a cilia. And we found some changes in BBS10 where we have changes in the way the cell divides, and there's a possible implication that it might have a knock-on effect on the ability to have this shortcut where you can form cilia as well. And we see some BBS10 being expressed during cell division and changes when we see BBS10 expressed there. So, yes, thank you.
So thank you, James. That was absolutely fantastic. It shows that all the research that you take part in actually has, there's actually something going on in the lab. But even more so, this is an area around, kidney research, which actually is really under-researched as far as BBS is concerned. So I think James's insights are going to be super valuable going forward. So thanks for that. Well, we've got time for probably just a couple of questions. Anyone have a question for James at all? Victor.
What promoter do you use to restore the BBS10 expression?
Sorry?
Promoter do you use? Do you use a promoter when you...
Yes, so we used a piggyback plasmid, so it just inserted hundreds of times within the genome. So I didn't actually do that experiment. We had a postdoc which did that for me, so I'm not sure of the specific promoter.
So it's not a specific promoter?
No, it just inserts hundreds of times, and you get very, very high expression.
Sorry, Chetan.
My question may be not as technical as Victor. I'll try and make an attempt. You mentioned healthy BBS10 genes are introduced. My understanding is BBS10 has mutated genes. What is the difference between a healthy and a poor cilia BBS10 genes?
So the BBS10 genes in patients who have changes in BBS10 will just have a change in the sequence. So I showed some earlier, but the healthy BBS10 gene will be what all healthy people will have a copy of BBS10. But with patients who have changes in BBS10, that's where you get the defects. So depending on the severity, patients might have a deletion where part of the gene just ends early, or a couple of pieces of DNA are changed, which means that when you make the protein, it's slightly the wrong shape, so it can't function as it needs to. So the healthy copy we put in is just the same sequence that any healthy person would have, without the changes that the patient with the mutations will have.
Tim?
There's a paper published earlier this year in Clinical Genetics about the type of mutation having a big factor in kidney disease and truncating mutations. And this is not something that ever occurred to me, but watching your BBS mice, are they all knockout, fully, like there's no BBS10 gene, or is there any work using models of truncating versus missense versus nonsense mutations? And is it the case potentially that BBS10 has more common renal failure because it's more common that those are truncating variants, not necessarily a BBS10?
Yeah, so that's a really good point. So the three lines we had, the one with the most severe phenotype has a truncating mutation, whereas the other two have genes which don't have, they have a copy without the truncating mutation. I think it's true that the truncating mutation is more severe. And the gene editing we did, where we gave the healthy cell line defective BBS10, we made truncating mutations to make it severe so that we could see and make sure we were seeing what happened. But we do have the ability to induce, we can try and tailor the mutation if we need to. So we could make very specific mutations and just change one thing to make it mild if we wanted to. We haven't done that yet, but it's totally on the cards. It's something that's we're definitely able to do.
All right, I think we'll move on from there. So thanks so much, James, for coming today.